Hello, my name is Michelle Bisiake. Today I will talk about a comparative analysis of the motility of CD4 positive and CD8 positive memory and naive T cell subsets. So a bit of background here. The study of immunosurveillance is a growing field of interest that works to understand the immune system's role in the protection of a host during tumorigenesis. Immunosurveillance is when an immune response is stimulated by the presence of cells undergoing transformation through the beginning stages of tumorigenesis. In this process, cells in the adaptive and innate immune system work to eradicate the growing tumor cells. In the adaptive immune system, T cells surveil and find foreign particles and then elicit an immune response, or they directly kill the cells, the tumor cells. Of T cell subsets, the CD4 positive cells are helper T cells whose effective function is to elicit an immune response in the presence of antigens and pathogens. CD8 positive T cells are cytotoxic T cells whose effective function is to cause a response against pathogens by secreting cytokines or defending the host by directly killing the T cells in the beginning stages of transformation. When activated, T cells proliferate and they differentiate into memory or effector T cell subsets. Memory cells have a much longer lifespan and have a high immune response upon reinfection. Therefore, they're more sensitive to antigenic stimulation. Naive T cells elicit an immune response but are less sensitive to antigenic stimulation. Scientists have mainly focused on immunosurveillance and T cell function from the perspective of direct cellular interaction. However, there are several more important questions that should be addressed on this topic from the perspective of cell motility. Because T cell motility directly affects their function and allows for these lymphocytes to perform their effective functions. So a few questions that we're going to focus on or have a bit in our mind are which cell subsets get to the tumor site and are more effective at attacking the tumors, or the growing tumors rather? Is migration of T cell subsets the reason why central memory is more effective in protecting the host against antigens compared to naive T cells? Of the CD4 positive and CD8 positive cells, which move more? And we'll work to answer these questions. In this image you see here, you see in the beginning slide labeled A, where it says elimination. This image depicts immunosurveillance, where you see the CD8 positive T cells, the natural killer T cells, and the CD4 positive T cells interacting with the cells that are beginning to transform. The CD8 positive and the natural killer T cells directly interact and directly work to kill off these cells that are transforming. The CD4 positive release cytokines that elicit an immune response. And so this one directly, this specific image directly models immunosurveillance. The methods that were done for this study was a blood sample was taken from healthy donors, and we worked to isolate out and store the peripheral mononuclear blood cells, or the PBMCs. We prepared the media, and then we isolated and expanded the immune T cell subsets, depending on which ones were necessary, from the PBMCs. And we labeled the cells and embedded them in a collagen matrix for a live cell microscopy study, so we can test and see the motility. Then we track the T cells movements computationally to obtain the rate of motility of each T cell. So here we compare the motility of total T cells in a low density and high density cell condition rather, CD4 positive and CD8 positive. In these graphs, you see overall that the CD8 positive T cells in a low density cellular condition with the most motile overall. We see the CD4 positive T cells in high density and the CD8 T cells in a high density condition have a similar motility. And then the CD4 T cells in a low density condition were much less motile than the CD8 in a high density condition. So we see different motility in this aspect of high density and low density conditions here. Of the CD4 subsets, so this is showing the effector memory, the effector memory and the central memory. The CD4 positive T cells in the central memory condition in high density were the most motile overall. And you see of all the different T cell subsets, the high density condition were the most motile in general. And this could be interpreted in many ways. It is really understood that in high density conditions, these T cells may excrete different or more cytokines, which could cause and stimulate more motility from these different T cells. And so that can be, that's a possibility as to why the high density conditions are more motile overall. And then you see that also the naive T cells are very motile, were also some of the most motile in these conditions. For the CD8 T cell subsets, we see that the effector memory T cells in the high density condition was the most motile overall, and the high densities in general were the most motile, which is a bit different than what we saw in the first comparison, where it was a low density was the most motile overall. We also see that the naive is almost as motile as the high density, and this can also be interpreted in many ways, as stated before, because of the different cytokines that can be excreted depending on the density conditions of these T cell subsets can cause a difference in motility rate. So in conclusion, we saw a few unexpected observations compared to what we thought would is previously seen biologically and what can be assumed by the functions of T cells. So naive cells were expected to be the least motile overall, however, they were much more motile than anticipated. For the CD8 positive T cells, the low density was the most motile overall, as we saw in the first comparisons. However, when looking at the subsets in the high density conditions of the individual subsets, they were the most motile. So that was a bit of a contradictory conclusion that was seen. And of the high density of the CD8 and CD4 T cells subsets, the high density conditions had a similar motility, which is a bit different than what's anticipated, because some people have assumed that CD8 T cells are much more motile than CD4 because of their cytotoxic effector function. And so it's a bit surprising that in a high that they have a similar motility, but it could also, there could also be many factors contributing to this. And this also has to be looked at with the consideration that only one biological repeat was done and several more have to be done for an accurate representation of this data and for accurate conclusions to be made. Some of the limitations of this study were that when the T cells were suspended in the collagen gel, they could not be left in for too long, or they would die. And so it was important, because of this, we could only look at them for about an hour and 30 minutes. And so we would look to maybe do it for a longer period of time to see their motility. And then also, if you can only observe this from a 3-minute time lag for it to be high throughput. I'd like to thank the NSF, the Institute of Nanobiotechnology at Johns Hopkins, the Wirtz Lab for their help, and my mentor, Adrian Johnson, and my PI, Dr. Denny Wirtz. Thank you.